So, now we all know how genetically engineered crops came into being and how they're made. But why am I telling you this? Why do I care? Who even am I? Well, I'm a member of Washington University in St. Louis's 2014 iGEM team. We're a group of undergraduate students who are exploring the realm of genetic engineering and will present our findings at an international competition in November. Over the summer, we've been working to take some of the first steps in fixing the nitrogen problem in agriculture. Baby steps. Baby steps, I will admit, but we're working on it. We've actually got 2 different projects going right now. In one lab, we've been working on getting the genes for nitrogen fixation, also called a NIF cluster, from a cyanobacteria to work in a foreign system. For our purposes, we've been using E. coli because it's widely studied, grows quickly, and is easy to engineer. So, we took the plasmid that contained the NIF cluster from our cyanobacteria and stuck it into the E. coli to see if it would allow the E. coli to fix nitrogen. We tested the nitrogen fixation activity with an acetylene reduction assay. Remember in that first video whenever I mentioned the super strong triple bond that happened between the 2 nitrogen atoms in an atmospheric nitrogen molecule? Well, acetylene has a super strong triple bond as well, so the enzyme that cleaves the triple bond in atmospheric nitrogen can split that triple bond in the acetylene molecule as well and form ethylene molecules. So, if I inject acetylene into the bottle with E. coli, then I can take a sample of the headspace with a gas chromatograph and see if there's any ethylene there. If there is a significant amount of ethylene, then bingo, we've got nitrogen fixation. So that's one thing we've got going on. In the other lab, we're actually working on designing a light-sensitive transcription system. Since the end goal is to get chloroplasts of plants to fix nitrogen, we want to alternate photosynthesis and nitrogen fixation. See, the proteins that are necessary to fix nitrogen are permanently inactivated by oxygen, which is a byproduct of photosynthesis. Super convenient since we also live in an aerobic world, but there you go. We want nitrogen fixation to happen in the dark when photosynthesis stops, so that there's less oxygen in there to wreck our nitrogen fixation system. Therefore, we are creating a transcriptional system that will turn on in the dark and turn off in the light. If we combine this system with our NIF cluster genes, then we can move this whole thing into another photosynthetic organism, such as a cyanobacteria. According to the endosymbiotic theory, cyanobacteria are actually the ancestors of chloroplasts. Basically, a cyanobacteria was just floating along, doing its thing, when a neighboring eukaryotic cell just swallowed it whole out of the blue. Talk about a rough day. Well, it wasn't that terrible because the eukaryotic cell became autotrophic, meaning it could make its own food, and plants just started popping up everywhere. Oh. Well, apparently it took millions of years for that last part to happen. But you get the point. So, based on this theory, the next step is to move from E. coli into a cyanobacterial system. That is way out of the scope of our little project, but that's what the future is. Hopefully we can live to see plants fix their own nitrogen and look back on this time fondly. Till then,